Hello, good morning, everyone. Good afternoon. It's a pleasure for me to be here and to present my work about the diet of the pond turtle, because I could not afford to travel to the US, and I think it's not really eco-friendly to take the plane for a week symposium. So it's a pleasure for me to be here, and I will present you a part of my PhD thesis. So first of all, the European pond turtle is the only native freshwater turtle of Switzerland. And there are two native subspecies in my country, which are Emys orbicularis and Emys orbicularis hellenica. The statue and the causes of regression. So it's one of the most threatened reptile in Switzerland. It's actually ranked endangered. And as always, it's due to human activities. The species was consumed in large quantities in the last century, and also because of the habitat loss and destruction for urbanization, agriculture, and so on. The Emys project, to go on a more positive note, I can't talk about the Emys project without talking to my turtle center. The turtle center is located in Switzerland. Oh, sorry. And here is our facility. It's quite new. We just moved there in 2008. And we are all volunteers there. We are active since 1994 now. And the first goal of the center was to rescue the turtle, because a lot of people release exotic individuals into the wild, and it could be really harmless for the Swiss biodiversity. So we take care of all these turtles. We have now more than 2,400 turtles at the center. And we are only open on Saturday morning. So here are some information if you want to go to have a look on our Facebook page. And in 1999, we launched the Emys project with the aim to reintroduce this species into the wild. And between 2010 and 2020, we released almost 120 individuals. But the question raised to determine or to know if this reintroduced species could be a threat for the endangered species. So if this species will feed on threatened amphibians, for example. So I decide as a part of my PhD to study the diet to determine if the European pond species is really a threat for other endangered species. So to do so, we have to study the diet as a function of time. So during the whole activity period, we did a capture session to collect feces, and we did field work from April to September. We choose to collect feces because it's a non-invasive method to get the data. And with this analysis, we could also determine the difference in diet between male and females, between adult and juveniles, and determine if there is a shift in the diet during the year. So usually, if you look at the literature, the diet of the pond turtle was studied using feces by direct observation or using a microscope, but there is a huge loss of information by doing so. So for example, if the pond turtle eats a worm, such as here, it's unlikely that you find any pieces of the worm in the feces. So we decide to develop a new method of global DNA amplification and metabarcoding using PCR primer to determine precisely the diet. So the fieldwork, this is the net we use for the fieldwork. They work very well without baits. And the only things you have to do when you do fieldwork, the weather has to be really nice, because the turtles are moving a lot when it's sunny. But if it's a rainy week, you won't catch any turtles. So we spend a week each month during the activity period, setting up the net on Monday and going back to the field each day to collect the turtle. We put them in individual containers, and the day after we collect the sample. We did not keep the turtle more than a day, so we release them the next day. We did some measurement. We took the sample to go directly to the lab to extract the DNA, and then we put back the turtle into the wild at the exact same place where we captured them. So in total, I collected 174 faces from 142 different individuals. So after the sampling, the lab procedure is composed of the grinding of the sample with the liquid nitrogen, the total DNA extraction of the sample. Then you have to do a lot of PCR tests to select a PCR primer targeting the plant, the vertebrates, and the invertebrates that could be found in the diet. Then you amplify all the sample, and you create the library for sequencing, and we use the Illumina Mini 6 to sequence all our data. And ultimately, you have to do to use bioinformatic pipeline to get the data and finally get the Emys diet. So the strength of the method, what we can say, it's that we get a great amplicon lens, and with that we could precisely determine the diet of the pond turtle to the species level. What we can say, our method is only qualitative, not quantitative, so it was not possible to determine the amount of food that was eaten. And unfortunately, nowadays with metabarcoding method, it's not possible to determine the stature of the prey. So we don't know if the prey was an adult, a juvenile, a larvae, or an egg, or if the animal was dead or alive. For the result, almost 90% of the prey were identified to the species level. This was really good because our method, we were very happy with the method we developed, and we identified more than 1,000 prey from 270 different species. So here are some of the invertebrate species we found. We found as well as aquatic invertebrates and terrestrial vertebrates. Maybe the pond turtle is going on land to feed, or maybe the animal just fall in the water and die in the water. We found also some vertebrates in the diet and a lot of different of plant species. So in total, on the 174 samples analyzed during the whole activity period, 90% of them contain plant DNA, 21% contain invertebrates DNA, and only 26% contain vertebrates. So to go more in detail, to determine the difference in diet by sex between the adult and the female, if you look at the metabarcoding analyze show us that 97% of the fecal sample collect on female contain plants, and 100% of the feces collect on male contain plants. The difference was not significantly different. We find the same for the invertebrate consumption. There were no significant difference between male and female. And the same for vertebrates, with a slightly higher number of vertebrates found in the male. So no significant difference. We were a bit surprised because at first we hypothesized that maybe the female will have a more carnivorous diet than the male because they have to produce the eggs. So the difference by maturity, we determine the diet for juvenile and adult, and both of them consume a lot of plants. a lot of invertebrates and a few vertebrates with no significant difference at all between the two diets. This was very surprising because in the literature you find a lot of time that the juvenile are more carnivorous than the adults, and they tend to become more herbivorous as they grow. But this was not the case in our study. The difference by period, we split our data set in two subsets, one for the reproduction period, the months of April to June, and one for the rest of the activity period, July to April. For the both periods, the consumption of plant was really high, the same for the invertebrates, but we got a significant difference in the consumption of vertebrates with more vertebrates consumed during the reproduction period. So this finding raised the question, is because the vertebrate prey are more available at this time, or is it because the species need like more energy to reproduce, to fight for female, to lay eggs, and so on? So the number of species consumed, we found 1 to 24 different species for each fecal sample. There is no significant difference between the number of species consumed between male and female, but a huge difference in the number of species between April and June, the reproduction period, and the rest of the activity period. We can look more in detail for each month with a huge amount of species consumed in June. Again, the same question as before, is it because the prey are more available or because the turtle needs more energy? Then to finally answer my main question, is the European bond a threat for other endangered species? So here you get the different species that are threatened in Switzerland, the statutes, and the number of time we find them in the sample and the percentage of it. So if you look at the vertebrates in orange, we find only a few vertebrates and 3 amphibian species, Bufo, Rana temporaria, and Pelophylax. But if you look at the number of time we found them is very, very low. And for Bufo, it was mainly at the beginning of the year in April. So we can hypothesize they feed on eggs or on tap food. But we don't know exactly. The only one we are sure it was an adult is Rana temporaria, because we found in the feces some bones. Then if you look at the plant consumption, the Nymphea alba were consumed a lot, more than half of the sample contains Nymphea alba DNA. It was really easy to determine that because a lot of seeds were found in the sample. So maybe we can say, I won't say this, the pond turtle is a treat for Nymphea alba, because I think it's more like a win-win relationships, because the pond turtle will feed mainly on the fruit, as you see with the seed in the feces. And a study in Spain demonstrates that the pond turtle disseminates the seeds through the pond, and that the seeds germinate better when they go through the digestive system. And finally, if you look at the purple species, they are the invertebrates. The number is really, really low. So we can, here is also a positive control. We find sometimes a hard part of the insect that could not be digested. So it's a positive control for the sequencing. So we can conclude that more than 85% of the species are not listed on the Swiss red list, so they are not threatened. And we can say that the impact on the impact is marginal on other endangered species, and that the feeding behavior of the pond turtle is really opportunistic and omnivorous. So the take-home message for this study, we demonstrate that the species eat a lot of plants. This was really important because on the literature, I found sometimes that the plant found in the feces were like eaten by accident. So we demonstrate that this is not the case because almost all the sample contain plants. The reproduction period was more diverse in diet and with a higher number of vertebrates. The feeding behavior is opportunistic and the number of endangered species found in diet is really limited. So the impact is marginal on the pond turtle, which is good for the reintroduction program. Like this, we can put turtle back in every pond of Switzerland. Thank you for your attention. I just wanted to acknowledge all the participants on my PhD. Without them, I couldn't undertake this work. Thank you for your attention, and I'm waiting for your question. Please be gentle. I just present my PhD today, so my brain is a little bit dead. So I see there are some questions. Are you reintroducing Emys into historical habitat or introducing into new habitat, suitable habitat? So we try as much as we can to reintroduce the pond turtle in historical habitat, but in Switzerland, in the region where you can find the pond turtle, 90% of the marshland have been destroyed for agriculture, for urbanization, and so on. So when it's not possible to find historical habitat, we do renaturation work to build up new suitable sites. The second question, did you have any issue getting turtle to defect, or did most defected during your 24 hours holding period? So the pond turtle is very nice, and it's a very good studied animal because almost all the turtle defect in 40 hours and sometimes less. As soon as you put them in the individual containers, a lot of them defected in the 5 following minutes. But I would say like 10% of them did not, but it doesn't matter because we had a lot of sample and we put them back in the water even if they did not give us a sample to study. Do turtles avoid invasive and stick to food they are used to, or do they eat more of invasive as they are more abundant? Unfortunately, we did not find invasive species in the diet, except one Pelophylax, but they are not targeting invasive species. I think in the pond where we studied the turtle, they feed mainly on native species, but pond turtle is only found in natural reserves, so these habitats are really preserved and they do not have a lot of invasive species. Did you capture and compare any red slider diet? Good question, thank you. This is my next step because I got a job starting next week, and the aim is to take care of all the invasive sorry, I'm reading as I'm talking, is to take care of all the invasive turtle species in Switzerland, and one of the goals is to study the diet and determine if there is difference or if there is difference between the Emys and the Emys orbicularis, to know if there is a concurrence in the diet. So maybe next year or in 2 years, I will present this work to the TSA meeting. So thank you for your attention, and I hope you enjoyed the talk. See you soon.